Hello and welcome to ATP Simplified. In this video, we're breaking down cellular respiration. Respiration is how cells get usable energy from food. The overall equation is kind of the reverse of photosynthesis. So glucose plus oxygen produces carbon dioxide, water, and ATP. And we'll go through 4 stages: glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation. Let's start with glycolysis. And the key is here, every organism does this part. So it happens in the cytosol, and no mitochondria or oxygen is required. One glucose gets split into 2 pyruvates. You make 2 NADH, and while you invest 2 ATP, you get 4 back, for a net gain of 2 ATP. This is substrate-level phosphorylation, which means directly making ATP by transferring a phosphate group. Next, the 2 pyruvates move into the mitochondria. Each pyruvate loses a carbon as CO2 and is converted into a 2-carbon acetyl group. NAD plus grabs some electrons and becomes NADH. The acetyl group attaches to coenzyme A, making acetyl-CoA. Since we started with 2 pyruvates, this happens twice. Now, acetyl-CoA enters the Krebs cycle, also called the citric acid cycle, which is inside the mitochondrial matrix. For each turn, you get 2 CO2, 3 NADH, 1 FADH2, 1 ATP. And again, since each glucose made 2 acetyl-CoA, the whole cycle runs twice per glucose. Now let's total things up so far. 4 ATP are made directly by substrate-level phosphorylation, and 10 NADH and 2 FADH2 are high-energy carriers that will head to the next stage, and all 6 carbons from glucose have now been released as CO2. Now here comes the big ATP payoff, oxidative phosphorylation. NADH and FADH2 drop off electrons at the electron transport chain in the inner mitochondrial membrane. As electrons move down the chain, their energy pumps H+ ions into the intermembrane space, creating a huge proton gradient. Finally, oxygen is the final electron acceptor, so it combines with electrons and hydrogen to make water. Those H+ ions now flow back into the matrix through ATP synthesis. This produces ATP, which is called chemiosmosis. For each NADH, you get 3 ATP. For each FADH2, you get 2 ATP. That's 10 times 3, which is 30 ATP, plus 2 times 2, which is 4 ATP. So we get 34 ATP from oxidative phosphorylation. Add in the 4 ATP from earlier, and you get a theoretical total of 38 per glucose. Though in real life, it's usually a little less due to inefficiency. But what if there's no oxygen? Cells can still make ATP using fermentation, which keeps glycolysis running. The purpose of fermentation is to oxidize NADH back to NAD+, so glycolysis can continue. So since NADH loses electrons and is oxidized, the pyruvates need to get reduced. In yeast, pyruvate becomes ethanol and CO2. That's alcohol fermentation, which is good for bread and beer. In human muscle cells, pyruvate turns into lactic acid. That's lactic acid fermentation, which doesn't release CO2. Alright, let's check your understanding. You can pause here and try this little quiz. Here are the answer keys. That's cellular respiration. If this helped, like and subscribe, and see you in the next video.